We'll wait for some more time, a couple of minutes more, and then we'll begin. Okay, I think we have waited enough. We can begin. So, welcome to the session 4 of Genome Editing and Engineering course, the live interactive doubt clearing sessions. This course is taken by Professor Utpal Bora of IIT Guwahati. And I am Sanket Mukherjee, a PMRF research scholar in the department, I mean, in the School of Biological Sciences in IIT Delhi. So initially, we will briefly discuss about the summary of week 4, and then we will get into few questions for this week. So essentially what was discussed in this week was targeted genetic modifications 1 and 2, and we observed that genes are modified. So what does targeted gene modification means? It means insertion or deletion of protein coding sequence at a particular locus in an organism's chromosome. So this modification is carried out in germline cells or in embryonic stem cells of an organism. So what is a germline cell? Anybody? You're clear with what is germline cell and embryonic stem cell? Okay, I would assume that you people know this. Germline cells are like the egg cells or the sperm. They are germline cells. So gene editing could be done at that stage or in the inner, cell mass of blastocyst. So that is during the embryonic stem cell stage. In these two stages, usually modifications are done. And two important methods are involved here, either gene knockout or gene knock-in. So what happens in knockout? You remove a gene of to see whether its absence will cause any effect in the organism's survival or a progression of a disease. So you do disease modeling and stuff like that. Gene knock-in is when you insert a gene and you want to see whether its overexpression would affect or it would cause some effect in the disease progression or the survivability of the organism. So that's what it is all about. So gene knockout is where in the wild type mouse, if you do it in mice models, in the wild type mouse, your gene of interest will be expressed, while in knockout model of your mouse, this gene of interest will be absent. It will be knocked out. It won't be expressing. So there are several strategies to do that. We'll discuss about it. But Essentially, this is the idea, this is the concept. When we are talking about knock in, then what we do usually in wild type mouse, we there is a gene that is being expressed, and at that gene locus where that gene is expressed, we add some other gene. This gene could be mutated gene, gene could be some other gene that we want to study about. So this is how knock-ins are created, this is why knock-ins are created. So coming to the next slide, what are the applications? So essentially, it is gene modification in the germlines of mammals. So there could be loss of function mutation in knockout, while gain of function in knock-in. So anybody would like to tell that what this loss of function and gain of function means?
Loss of function means not expression. No, the gene is not expressing and the function is not done. Gain of loss of function. Gain of function means the gene expressed and the protein is doing the function.
Yeah, that is correct. So essentially, if you're studying about cancer models, I'm just giving an example here, then loss of function of, let's say, tumor suppressor gene would enhance the development of cancer. Similarly, gain of function in oncogenes would cause, again, enhancement in development of cancer. So these kind of studies, they are done using knock-in, knockout models of mice or any other organisms that you would be, taking as a model organism. Now, a specific gene, the function of the specific gene is studied uh, using this knockout and knock-in system. So if you knock out a particular gene, and the effect of that gene could be analyzed, because if that gene is absent, there would be some consequences because of the absence of that particular gene. Then there is creation of transgenic animals. Transgenic animals means these are animals that have different genes. Even chimeric animals are developed using this kind of strategy. Development of disease models, just as I said, like cancer models or even diabetic models, these are all created using knock-in, knockout mutations. And some medical therapies are also involved as one of the applications. So in 2007, these three people, Capecci, Evans, and Smithies, they discovered, um, you know, they had the discovery of the principle for introducing specific gene modification in mice by use of embryonic stem cell. So essentially, gene targeting gene modifications, that is what they discovered. We will see each of them, each of their contribution. So in 1960s, Oliver Smithies, he established that homologous recombination causes allelic variation in human haptoglobin genes. So this has been described in detail in the lecture. We are just doing a summary here. So similarly, Michael Wigler and Aikes, they in 1977, they did a phenomenal job in regaining the enzyme thymidine kinase gene in mammalian cell culture. And the functional copies of this gene was introduced from a herpes simplex virus. Uh, and then this uptake of this gene was done using phagocytosis. However, the efficiency was low. Now, in improving the efficiency, Mario Capecci, he was involved. He solved the problem of this low efficacy by devising a method to introduce DNA directly into the nucleus, bypassing the cytoplasm and lysosome through injection by a tiny glass pipette. Even nowadays, we have gene gun which performs the same function. So even now we have certain techniques which are inspired from this. So for insertion of genes, we still perform something like that, and he improved the efficiency slightly better. Then what happens? Martin Evans, he so they did genetic alterations. I mean, Capicci and Smithies, they carried out genetic alterations in somatic cell lines, and Evans, he did it on embryonal carcinoma cell lines, and he enabled this work to be carried out in germline cells as well, apart from somatic cells. So Evans, he worked on embryonal carcinoma cell lines derived from mouse testicular carcinomas, and these cells induce different. These cells, they can be induced to differentiate into multiple tissue types. What is this called? If a cell can differentiate into multiple tissue types, what is it called?
Pluripotent stem cells.
Pluripotency. Okay, so he observed pluripotency first in the embryonic stem cells. So through cell culture, he characterized that teratoma-derived cells of mice strain have high frequency of tumors harboring totipotent cells. So we'll come to it. So essentially what happens. Okay. Essentially what happens is, here you can see in the human fetus there is blastocyst stage, and in the blastocyst stage we take the inner cell mass, which contains the pluripotent cells, which can differentiate into multiple cell types. So this was an important thing. While once these cells are differentiated, they can no longer be, they can no longer form any kind of cell. So let's say if pluripotent cell, it forms an immune cell, or let's say hematopoietic cell. So that hematopoietic cell cannot form neural cells. So that differentiation, once the cell has differentiated, it cannot go back usually. So this is what was observed. Then also he created chimeric mice. So chimeric mice means like there are uh, two different kind of mice, I mean two different kind of cells injected into the same mice, and we form a organism that is, we call it as chimeric, when it has two different genes, or two, it could be a lot of things actually in that way. So he injected the embryonal carcinomal cells into the mouse blastocyst, which were then implanted into the foster mother. And then, since it had some carcinomal I mean cells, and some normal cells, both were present. Therefore, it was uh, a chimeric model, right? So when the EC were injected with retrovirus, the retroviral DNA was detected in both somatic and germline cells of the chimeric mice. Anyway, this is how chimeric mice is produced. So you take the embryonic cell from one mother, and you transplant it into the embryonic cell of another mother, and that's how you create chimeric organisms. So we talked about serine tyrosine recombinase earlier. So as we said that serine recombinases, one of their characteristics is the rotation and ligation. That is one of their characteristics. And tyrosine recombinases, it involves formation of a holiday junction intermediate. So we discussed about it previously. Then we have something which we discussed in this week was about Cre recombinases. So Cre recombinases are usually used for creating conditional knockouts. So there are like LOXP sites. In between, we have the target gene. So when Cre is expressed conditionally, at that time, this LOXP, these flanking regions, they cut the, they are removed basically from the site, and that's how we create conditional knockout systems in Cre LOXP. So why would conditional knockout be important? Anybody?
To cut out the targeted gene which we want to see the loss of function.
That is correct. But why can't I just do knockout? And why do I need conditional knockout?
Because it may, simple knockout means it can happen anywhere. Conditional means we have to
Correct.
give some specific.
Right. So we can make tissue specific knockouts. Okay. We can also make time specific temporal knockouts. So let's say if you create some knockout that causes some uh, defect in or fatality during birth. So you want to keep this gene intact, working during birth and in the initial stages of infancy, but later you want to knock it out and see whether the same gene has some important role even in the late phases of your life. So something like that. When you need a condition to do, to knock out a gene, you will use these kind of knockout system. Similarly, we have the FLP FRT recombinase system. Here also, the FRT, in between there is FLP and the gene, which is then knocked out, which is excised essentially. And then we have the TET on and TET off system, which is again one of the most used systems for knockouts. So what happens if you have a TET off system? Then if doxycycline is given, doxycycline is a class of antibiotic. TET on and TET off. So TET stands for, what does TET stands for? Tetracycline.
Tetracycline.
Okay. So doxycycline is also a drug, or it's an antibiotic which is a part of this tetracycline group of compounds. So if you have a TET off system and you introduce doxycycline, then the gene expression will be lowered, it will be stopped. But in a TET on system, if you introduce doxycycline, then the gene expression will be induced, or gene will be expressed. So what happens in a TET-off system? If you're giving doxycycline, gene expression will be stopped. If you're not giving doxycycline, gene expression will happen. In a TET-on system, if you're not giving doxycycline, gene expression will not happen. But if you're giving doxycycline, then gene expression will happen. This is essentially what happens. And these are controlled by TET control transactivators and reverse TET control transactivators. These are essentially which controls the system. So they are also used for developing models uh, where we see where we want to fine tune our expression of the gene expression using something else. So for designing vectors, which was discussed in the lecture 2, so what needs to be there? There should be a selection marker gene, a positive selection genes, like it is usually an antibiotic selection gene like neomycin, puromycin, hygromycin. So what happens if this antibiotic is, when you grow these cells which have the knockout or the knock-in, whatever, when where you have done the gene modification, when you grow these cells in presence of these antibiotics, they will survive, the rest will die. Clear? Is it okay? Okay. So in negative selection, you usually choose, this is done to prevent prevent any random integration, because usually when we design these vectors sometimes they are prone to any random integration of DNA, or there could be incomplete knock-ins, knockout being generated. So to rule out those kind of problems, those kind of gene modifications which are incomplete, we usually use negative selection. So thymidine kinase is one of them. So whenever there is random integrants, then there is negative selection through treatment with kylovir. So this thymidine kinase gene, it causes phosphorylation of these compounds, which inhibits the DNA synthesis. So there is both positive selection and negative selection to make sure that only the cells that have the proper gene which we require, they only survive. So again, in vectors, there are two types of vectors. One is replacement vector, another one is insertion vector. Just to explain you very briefly, a replacement vector, both they work with homologous recombination. So what happens in a replacement vector, your target gene is inserted, but it replaces some sequence. That is what happens in a replacement vector. But what happens in a insertion vector, it inserts its sequence. It does not replace, but rather insert its sequence. So this is the two difference between insertion. I mean, it's the difference between replacement and insertion vector. So any examples of insertion vector? Does this remind you of something? I think it's even written in the slide. Lambda phage, that can be used as an insertion vector also. So, okay, subtle mutations, they can be introduced by either replacement or insertion type vectors. Now, for introducing the subtle mutations, there are three approaches. One is the hit and run approach, tag and exchange approach, and the recombinase based approach. So what happens in hit and run approach? There is an insertion vector, and then that will insert the target sequence, and then we will have positive selection because we will have our selectable markers present there, and then there will be intrachromosomal recombination that will introduce that mutation. That is one way. Another way is tag and exchange approach, where what happens, there is homologous recombination with one vector, then there is homologous recombination with another vector. So during this process, your gene of interest, that when it forms an homologous recombination. So here, uh, when it forms a homologous recombination, this will be swapped, and then there will be side directed recombination in the chromosome, and then this will be inserted in the chromosome. So that is another approach. And the third approach is the recombinase approach, like we talked about the Krelog system. That is also one approach for introducing subtle mutations. And in the end of the lectures, we discussed about creation of knockout knock-in mice. So we have embryonic stem cell culture. We culture these cells, and then we construct the targeting vector, which could be insertion vector, replacement vector, any kind of vector, whatever we need to prepare. We need to construct these target vectors, and then there will be transfection. So what is transfection? You know,
Introducing a new gene into cells
yes, correct. So
or
when we
any
do
any
this in,
plasmids.
yes, right, plasmids, vectors, new genes into cell, this process is called transfection for eukaryotic cells. So that is what transfection is. So you have constructed the targeting vector, then you need to insert this targeting vector into the cells, embryonic stem cells, and then you will allow homologous recombination to happen, and then you will perform selection, positive and negative selection, to make sure that only the cells that have been transfected, who have been transformed, efficiently, only those cells, they only survive, the rest die. So that is the process. So we'll discuss about few questions. So is it clear so far? Okay, I hope that is. Then let's begin. Who established that homologous recombination causes allelic variation in human haptoglobin genes? Is it Oliver Smithies, Mario R. Capecci, Martin J. Evans, or Michael Wigler? Anyone would like to answer?
Is it Oliver Smith?
You're correct. You're absolutely right. So that is the answer. The inner cell mass localized at one pole of the blastocyst is the source of dash cell lineage. Is it unipotent, multipotent, pluripotent, or totipotent? Pluripotent, yes, you're right. Pluripotent is the answer. So there could be multipotent cell lineages also. Like we have, the hematopoietic stem cells. Hematopoietic stem cells, they can differentiate only into the hematopoietic lineage. So those kind of cells, stem cells in particular, they are called as multipotent. They are not pluripotent. They cannot form any kind of cells, but they can form all the kinds of blood cells, whichever, of the hematopoietic lineage. So that is the difference between pluripotent, multipotent, if anybody is confused on that regard. So recombinase, it belongs to dash class of site-specific recombinases. Is it serine, tyrosine, leucine, or methionine?
Tyrosine.
You are correct. You are absolutely correct. It's tyrosine recombinase. Cre-Loxp system, it consists of two 13 base pair dash that flank a central sequence of 8 base pair, which determines the directionality of the Loxp site. So is it a palindromic sequence, AT rich region, GC rich region, or reverse sequence?
Palindromic
Any,
sequence.
that is correct. That is correct. It's palindromic sequence is the right answer. In a conditional knockout strategy, LOXP sites can be transferred around the exon of a gene, the intron sequence, 3' end of DNA, or the poly A tail.
Exon of a gene.
That is correct. So if this is the exon, means the part of the gene that is expressed, right? Is that known to everybody? Everybody knows this?
Yes, sir.
Okay, so if you introduce LOXP sites in around the exons, then this whenever you will have the Cre will be expressed, then this exon will be removed. Clear? That is the idea. That is how you will have the knockout. Okay. Then hit and run approach is used to induce what? Deletion, inversion, transcription, or subtle mutation. Any
Subtle
answers?
mutation.
That is correct. That is correct. Which one of the following options is incorrect regarding the generation of an incomplete gene knockout? Alternative or cryptive promoters, constitutive promoters, differential splicing, or read-through transcription? Did you understand the question? In which of the following conditions incomplete knockout is generated? Alternative or cryptive promoters, constitutive promoter, differential splicing, or read-through transcription?
Constitutive promoter.
So you know what this is, right? You people must have understood. Just for the sake of everybody's understanding, I'll try to explain each on one of the options. So what happens if there is alternative or cryptic promoters? So there might be some other promoters. So you might have knocked out one promoter, but there are some alternate promoters that are present for the expression of the gene. So what might happen? There might be residual gene expression. So that is an incomplete gene knockout, one of the conditions of incomplete gene knockout. Another thing that could happen is differential splicing. So what happens in differential splicing? It is possible that due to the splicing mechanism, there is a differential splicing that happens, and the selectable marker which we use as for positive selection or negative selection, that gets spliced out. In that case, we have an incomplete knockout. Or we could have read through transcription. What happens that we introduce some sequence to prevent the gene expression, but what happens that even after that sequence, that gene is there, so it gets expressed. So there is the transcription is read through, and some part of the protein is produced, and we have some expression. So maybe the targeted gene is not completely knocked out in that case. So these kind of things result in incomplete knockout. Then we have a recombinase. It carries out translocation when LOXP sites are in the same direction, opposite direction, on complementary strands, or on the same chromosome. Anybody?
Is it in the opposite direction?
LOXP is present in the opposite direction.
Yes, I think. I'm not sure.
Okay. Anybody else?
Translocation means removal of that particular gene, knocking out.
Translocation is, you know, literally means what it is written. So it is removed and it is placed somewhere else. That is what is translocation.
In the same direction. Then it is in the same direction.
The same direction. Okay. Anybody with any other answer? Okay, I will see if I have something to show you here. Translocation is when what happens in a translocation. Uh, so one gene to another gene, that kind of thing. You know about jumping genes, they are called translocons, something like that. Such kind of things happen. We are not excising the gene out. See, that is not the criteria here. So it's essentially translocated. That happens when it is present on the complementary strands. Let me go back to the recombinase part. Okay, here it is not described. Let me see if I can find something for you to show you. Okay, just wait for a minute. I'll be just sharing. Just a minute. Can you see the slide? Can everybody see the slide?
Yes, sir.
Okay. So so what were the options that we had? Just for, so it could be in the same direction, it could be in the opposite direction, it could be on the complementary strands or on the same chromosome. So if they are in the opposite direction, then what recombinase does, it inverts the gene, right? So that this is how it works out, as you can see in the figure A. Now if they are present in two different chromosomes in complementary fashion, then there could be recombination between these two chromosomes. So what would happen? There is translocation. So one will be chromosome 6. This is chromosome 6, then chromosome 3 together. And here we have chromosome 6 and 3. This kind of recombination is happening when it is present in two different chromosomes. and deletion happens when they are in the same direction. So deletion or knockout, when we do, then it is present in the same direction. Is that clear with everybody? Okay, so shall we call it a day then? Is everybody okay if we end the session here?
So I have a few questions.
Please go ahead.
So this Cree system is derived from the viruses, right? Will it not
Mhm.
elicit any antigenic properties in the host?
No, actually it doesn't. Not that I'm aware of. So I'll have to look into it, but not that I'm aware of that it creates any antigenic, I mean, immune response as such.
Okay. One more is what about the repair mechanism? We are inserting a new gene means the repair mechanism in that particular organism, will it not take any further actions to remove that?
Yes, yes, yes. That actually happens, and this is the reason why the efficiency is so low when you go for these kind of experiments. So the efficiency is so low because of these kind of problems that come in. So yes, that could happen.
And one more, sir. Actually, we are introducing this into blastocyst, right? Again, this is, we are again giving it to foster mothers. And will the embryo will survive, or is there any chances of miscarriage?
Survivability is also low. So when we do stem cell, again, this is not a part of this course, but probably in the end you will have something about stem cells also. But in stem cell research, uh, we usually do not use embryonic stem cells for this particular reason. So if you mess with embryonic stem cell, there is a lower chance of survival. That usually happens. But when we are working with mice models who have multiple litter mates and stuff like that, things are not, we are not that much concerned. But definitely, at least in terms of humans, there is some ethical issues that come up. That's why we produce induced pluripotent stem cells. So here we add some genes that will make a somatic cell to de-differentiate into a stem cell. So that's a separate concept, but yes, you said that is right. I mean, during production of chimerics also, there is a possibility that there could be fatality essentially.
Thank you so much.
Any more questions? Anything else? Okay, so shall we, if it is okay with all of you, you people can leave and we can end the meeting here. Thank you very much for joining in. We will be meeting on the next week, same time. Thank you.